Hello, this is Anuja Matthew. I thought it would be fun to recap on lectures for the week of March 28th and answer some questions. So this is my personal email below. Feel free to leave a comment on the video or even email me if you have any questions. I'm learning as we go forward. So I'm a cellular immunologist. My training has been trying to understand immune responses to virus infections. The two viruses that I study are dengue and Zika virus. I talked a little bit about coronaviruses, how they are positive sense RNA viruses, and how they have caused a pandemic that we are currently experiencing. I talked about basic virology, how a virus enters a cell, how viruses can be classified based on the genetic material, and how maybe even the infections that they cause. And in my last lecture, I talked about immunology. How do you generate a T cell response? How do you generate a B cell response? Why are they important? And I'm hoping in the next week to go into a little bit more detail about T and B cells and some additional data on a coronavirus immune response. Thanks for your attention. So a couple of questions on my lecture on viruses. What's the difference between RNA and DNA, and is there an advantage to be a positive sense RNA virus? So both RNA and DNA are made up of a chain of nucleotide bases, but they have slightly different chemical properties. DNA is a blueprint for all genetic information contained in an organism. That includes every cell in your body as well. DNA is typically found in the nucleus of a cell. A small amount of DNA is also present in a structure called the mitochondria. And RNA basically uses the information and converts the genetic information contained within DNA to form proteins. So if you have a skin cell, every protein that makes up a skin cell goes from DNA to typically messenger RNA. The messenger RNA goes from the nucleus of the cell to the cytoplasm, and each messenger RNA gets translated into a specific protein for that cell type. So viruses, as I mentioned in my lecture, also contain genetic material. They're typically RNA or DNA. And there's all kinds of versions. There's single-stranded, double-stranded RNA and DNA viruses. So a virus, such as a coronavirus, as I mentioned before, has spike protein on its surface with genetic material inside the virus. So the sole purpose of a virus is to infect a cell, come in, use the machinery of the host cell and produce more viruses. So more viruses come out compared to the amount of viruses that go in. So coronaviruses are positive sense RNA viruses. And as I had mentioned before, their genetic material is the same as the messenger RNA. So they come in, their genomic material is uncoded. They have an enzyme called RNA-dependent polymerase in which they are directly able to form many of their proteins. So this is a much quicker way of coming in and replicating and trying to get an infectious virus formed together. So I think there's an advantage of being a positive sense RNA virus because you don't have to go to the nucleus of a host cell to replicate, and it's faster to produce more infectious virions out. So another question was, what's the difference between a virus and a bacteria? So bacteria and viruses cause many diseases, and they are too small to be seen by the naked eye. A bacterium is a single but complex cell, and it can survive on its own. So it doesn't need a host cell to survive. Most bacteria are not harmful. So we have millions of bacteria inside our body and on our body, in the skin, and especially in the gut to help digest food. Bacteria can cause severe illness, but there's plenty of bacteria that are harmless. Viruses are much smaller than bacteria. So here is a scale showing the size of a human versus, say, a chicken egg versus an animal cell. So cells that contain machinery to form, say, skin cells or lung cells or any kind of cell in your body. And these are organisms such as a bacteria, which is bigger than, say, a virus. And as an example, this is a flu virus. Viruses are smaller. They are not cells, and they do need a host cell. So they need either an animal or another bacteria or a human to multiply and replicate. I hope this is clear. So the last question I was asked, are zoonotic infections on the rise? So what's a zoonosis? So a zoonosis is an infectious disease caused by bacteria, viruses, or parasites that spread from animals to humans. So zoonotic disease pathogens such as rabies virus has been causing outbreaks in humans for thousands of years. So this is not anything new. But in the last 50 or 60 years, the rate of zoonotic disease, the incidence and prevalence has been on the rise. So examples of zoonotic diseases include West Nile virus that are transmitted by birds and crows, Ebola transmitted by fruit bats, avian influenza that caused the H1N1 outbreak transmitted by birds, Zika virus, plague, historically from rats, etc. So I'd like to end the session this week on the Q&A. Next week, I hope to upload some basic immunology videos on T cells, immunology videos on B cells and antibodies, and maybe hopefully get into some practical applications. How do they really diagnose a coronavirus infection? What methods do they use in the lab? Why has it been challenging to get diagnostics up quickly? That might be something of interest to this crowd. Thanks again for your attention. This is Anuja Mathi.